The overall goals of this procedure are to assess endocytic activity of liver cells, followed by isolation of a pure population of viable liver sinusoidal endothelial cells that can be cultured in plastic dishes. This is accomplished by cannulating and excising the liver of a live animal and flushing the liver with a salt-buffered solution to clear out the blood. The cells of the liver are then exposed to labeled ligands for internalization. The next step is to digest the liver with collagenase to produce a mixture of liver cells in suspension. Next, differential centrifugation with Percol gradients is used to separate SECs from stellate cells, small hepatocytes, remnant blood cells, and cell debris. The final step is to separate SECs from Kupffer cells by adhesion on glass. In the end, viable purified SECs are obtained that can be assayed for ligand uptake and/or be cultured for downstream applications.
This method can help answer the key questions in the field of liver biology, such as the role of the liver endothelial cells in the clearance of macromolecules in blood, or the responses to different physiological conditions such as liver injury, fatty liver disease, cirrhosis, fibrosis, and aging. Unlike other liver cells, immortalized differentiated liver sinusoidal endothelial cell lines do not exist. So these primary cells must be derived from the animal directly.
The procedure that we're going to show you today involves the purification of liver sinusoidal endothelial cells without the use of capital equipment like centrifugation and elutriation. The procedure that we'll show you involves the harvesting of cells on a short-term culture basis, where we're not going to use sterile technique throughout the whole procedure. However, if you want to do long-term cultures, you would use sterile technique and use a biosafety cabinet. Visual demonstration of this procedure is critical because placement of the catheter, along with tightness of the sutures, as well as taking the liver from the animal without cutting the liver, as well as disengaging the catheter, is critical.
Prepare for the experiment by weighing out the collagenase and setting it on ice. Then warm and oxygenate the TBS and prepare the required tools. Next, select a 200-gram rat and deeply anesthetize it in an isoflurane chamber. Confirm the anesthesia by wetting the ventral abdomen with 70% ethanol. If there is no response, proceed with opening the abdominal cavity using scissors and forceps. For best results, the following steps through blanching of the liver should only take a few minutes. Locate the portal vein and place 2 sutures beneath the vein just above the mesenteric branch. Now cannulate the vein with an 18-gauge catheter. The needle plugging the catheter should go several millimeters beyond the loop of the thread. Trigger the needle's release and tighten the suture with 2 overhand knots, also known as either granny or square knots. Begin flushing the blood from the liver with TBS for no more than 10 minutes. Sever one of the major arteries. While the liver is flushing, excise the liver by cutting away the GI tract and connective tissue. Place the excised liver over a funnel that allows recirculation of fluids, but do not yet recirculate the fluids. To 50 milliliters RPMI media in a small beaker, add to a final concentration 0.05% BSA and 0.01 millicuries radio-labeled heparin or other appropriately labeled ligand for endocytosis. Attach this beaker to the apparatus to allow for oxygenation. Turn off the pump, switch the inlet tubing, and then turn on the pump at 20 milliliters per minute. As the labeled RPMI approaches the liver, turn off the pump and allow excess fluid in the funnel and tubing to drain. Place the drain tubing into the media beaker to allow recirculation of labeled media, and then restart the pump. Allow fluids to recirculate through the liver for no more than 1 hour. During this internalization step, be sure the temperature in the liver is stable by covering it, and prepare the collagenase for digestion. To terminate endocytosis of the ligand, the liver is reflushed with TBS for 2 minutes to clear the liver sinusoids of unbound ligand in preparation for the collagenase digestion. During the 2 minutes of reflushing with TBS, dissolve the pre-weighed collagenase A in buffer 2 to a concentration of 0.45 micromolar and filter the solution. Immediately after flushing the liver, turn off the pump and switch the fluids to flush in the collagenase A. Begin flushing with recirculation at the same flow rate for up to 15 minutes. After the collagenase A perfusion, remove the catheter and transfer the liver to a dish containing buffer 1. Using forceps, peel back the capsule of the liver. And shake the cells out in the liquid. As the liquid becomes opaque, pass the cells through a 100 micron mesh, followed by a 30 micron mesh, and then into a 50 milliliter conical cylinder at room temperature. Add more buffer 1 to the dish and mechanically dissociate more liver cells, then transfer them to the filter. Continue this process until no cells can be dislodged with reasonable shaking. Begin by washing the cells. Centrifuge the cells at 150 times G until they are pelleted. Then, transfer the supernatant, containing non-parenchymal cells, to a 50 milliliter conical on ice and resuspend the pellet in 40 milliliters buffer 1. Repeat the wash process twice in buffer 3 to make pellets that are at least 97% hepatocytes. To obtain a population of SECs that is at least 95% pure, centrifuge the conicals of supernatant at 200 times G for 10 minutes at 4 degrees Celsius. Then, discard the liquid and resuspend all of the cell pellets in 5 milliliters of RPMI BSA at 4 degrees Celsius. Pool the cells into 2 tubes and fill the tubes with RPMI BSA to 35 milliliters. Now centrifuge the cells at 100 times G for 3 minutes and transfer the top 25 milliliters of supernatant containing the SECs to a 50 milliliter conical on ice. Then, resuspend the pellet in the remaining 10 milliliters of media and add 25 milliliters of cold RPMI BSA solution. Repeat the wash step once to make 2 more collections of supernatant with SECs. Now centrifuge the supernatant collections at 200 times G for 10 minutes at 4 degrees Celsius. During the centrifugation, fill 3 conicals with 20 milliliters of 25% percol in PBS. Put them on ice and set up a gradient by underlaying 15 milliliters of 50% percol in each tube. Resuspend the cells in a total of 30 milliliters RPMI with BSA, combine them, and then carefully overlay each gradient with 10 milliliters of the combined cell suspension. Centrifuge the gradients at 900 times G for 20 minutes at 4 degrees Celsius. Because SECs and Kupffer cells are at the 25-50% interface, aspirate from the top down to near the 25-50% interface. This removes the stellate cells. Now collect the cells in the 25-50% interfaces and transfer them to a conical with cold RPMI to a final volume of about 40 milliliters. This dilutes out the percol. Centrifuge the SECs and Kupffer cells at 350 times G for 10 minutes at 4 degrees Celsius. Resuspend the pellet in about 10 milliliters of pre-warmed unmodified RPMI. Transfer the suspension to a petri dish or crystallizing dish and incubate the dish for 15 minutes at 37 degrees Celsius. After the incubation, gently agitate the dish and collect the SECs in the supernatant. The Kupffer cells will have adhered to the glass, and the SECs can be cultured on fibronectin-coated plates with modified RPMI media. Cell lysates from cells that adhered to the dish and those that remained in suspension were separated by SDS-Page and probed with anti-CD163 antibody. Kupffer cells, which contain the CD163 marker, were separated from SECs by attachment to glass. Cells that did not adhere to the glass were plated on fibronectin-coated plastic 6-well dishes and incubated overnight with RPMI supplemented with 0.1% BSA and pen strep. Under phase contrast light microscopy, SECs exhibit a very flat cytoplasmic morphology, reminiscent of their in vivo architecture. A monoclonal antibody against both isoforms of stabilin-2 receptor, a marker of SEC differentiation, was used to probe hepatocyte lysate and SECs collected using the outlined protocol. As expected, the SECs were positive for stabilin-2. In prior experiments, injected heparin labeled with a biotin tag on the carboxylate group of the heparin disaccharide was conjugated to iodinated streptavidin. During the 30 minutes post-injection, the liver was the primary organ that cleared the labeled heparin. Using the procedures in this video, we show that the vast majority of labeled heparin is internalized by SECs in contrast to hepatocytes. These internalization events occurred within an intact liver, in which cells are in their native state.
So after watching this video, you should have a good understanding of how to purify liver sinusoidal endothelial cells from a live animal. And it's important to use these cells within 24 to 48 hours, as liver sinusoidal endothelial cells lose their distinctive characteristics in a very short time in culture.
All right, let's talk about the liver's unsung heroes. Sinusoidal endothelial cells. So, you know your liver is a filter, right? But what's the filter inside the filter? It's your body's metabolic HQ and main blood filter. It's basically a bustling cellular city.
Okay, check this out. Most of the liver is made of one type of cell, hepatocytes.
But that other 15%, that's where the real specialists are. Today, we're talking about SECs.
So what do? They form the walls of the liver's blood vessels, acting as gatekeepers.
And their superpower is something called fenestrae. Just think of them as super tiny, precise holes.
We're talking incredibly small. These pores create an ultrafine sieve for your blood.
So small stuff gets through, like nutrients. But bigger things, like blood cells, nope, they're blocked.
But wait, there's more. SECs aren't just a passive filter. They're also an active cleanup crew.
How do they clean? With these special tools called scavenger receptors that literally grab waste.
Think of it as a team effort. SECs get the small junk, while Kupffer cells handle the big stuff.
And one of their main targets is this molecule called hyaluronan.
Why does that matter? Well, if hyaluronan builds up, it's a huge red flag for liver disease.
So if these cells are so critical, why are they so dang hard to study?
Here's the problem. They're super delicate and almost impossible to keep alive in a lab.
In culture, they basically forget who they are. They just become generic cells.
I mean, look at this. In just a day or two, those crucial fenestrae just disappear?
This makes studying them long-term a massive headache for scientists trying to find new therapies.
So the SEC is the liver's unsung hero, a delicate sieve and an active cleanup crew.
Which leads us with a big question. How can we control these gatekeepers to unlock new therapies?